Hello, my name is Jonathan Schneck, and I'm a professor in the Department of Pathology here at Johns Hopkins School of Medicine, and I have my labs in the Institute for Cellular Engineering. What I'd like to talk to you about today is some new work that's coming out in JCI, spearheaded by a graduate student, Yenling Chu, on the genetic regulation of HIV-specific T cell polyfunctionality. But before we talk about the work, let me just talk to you in general about T cell and normal viral infections. So ordinarily, when we get infected with a virus, what happens is that over a time course of 7 to 10 days, T cells get activated. In the process of getting activated, they proliferate a lot, and they acquire effector functions that allow them to eliminate the viral infection. And if all goes well, this will be a sterilizing event, and you'll be viral-free within 2 weeks time period. However, that doesn't always happen, and in the setting of chronic viral infection, the virus is not fully eliminated, and the T cells get chronically stimulated, as in HIV and hepatitis C, as well as in cancer, and those cells become dysfunctional or exhausted. We were particularly interested in the genetic regulation of the dysfunctional cells, and that's the work that Yenling started. That's the work that we're reporting today. What was required for this was the development of a model system where Yenling was able to short-circuit that chronic activation and chronic persistent antigen to set up the model system so that we could look at the genetic regulation. And sure enough, he was able to do that using influenza-specific T cells.
To study the genetic regulation of T cell polyfunctionality, we used T cells from healthy donors and layer on autologous dendritic cells. We co-culture T cells with dendritic cells labeled with different level of peptide antigen. And what we found was that the T cells that was induced with the highest amount of peptide antigen were dysfunctional and lose polyfunctionality, while there was an optimal amount of antigen on dendritic cells that can induce T cells with the most polyfunctionality. As shown here in the pie chart, the red portion, that means this percentage of cells are capable of doing 5 effector functions simultaneously. And we were also being able to perform a microarray analysis on these antigen-specific cells, and we found that there is a genetic signature of T cell exhaustion were enriched in the T cells that were dysfunctional and induced with high dose of antigen.
When we got the microarray results, we were really very, very excited because it indicated that some of the pathways we expected were indeed upregulated in the dysfunctional T cells. One of those pathways that was novel, though, was to see that the MAPK kinase pathway was implicated in the dysfunctional T cells. So this was a big surprise to us. We expected the MAPK kinase pathway. It's a general regulator of T cell activation. We didn't expect to see it just in the polyfunctionality aspects of the T cell activation, and indeed that's what the case was, is that we saw it just in the polyfunctional T cells. And even more so, one of the proteins that was upregulated was SPROUT-E2. SPROUT-E is a protein that hasn't been very much studied in the immune responses. We actually know about it from Drosophila. But SPROUT-E2 is known to be a master negative regulator of the MAPK kinase pathway. And so we're very excited when SPROUT-E2 was implicated in the negative regulatory aspects of the decrease in polyfunctionality. What Yenling did was then tested it in the model system. He saw that when he reversed SPROUT-E2, indeed we saw upregulation of polyfunctionality. And even more importantly, when we looked into the HIV cells, when we got cells from HIV patients and downregulated SPROUT-E2 expression, we increased polyfunctionality. And by targeting 2 pathways, one already known and this one that we just described here in this article, we're able to completely reverse the defect in T cell, the dysfunctional T cells. Thank you for your time, and I invite you to look into the paper and read the paper in JCI.